So for physiology, we'll be discussing about the cell. First is the physical structures of the cell. We have the cell membrane, the cytoplasm, and the organelles. So for the cell membrane, also known as the plasma membrane, it envelopes the cell. It is a thin, pliable, and elastic membrane which has 7.5 nanometer thickness. And it is composed by lipid bilayer. This is lipid soluble substances can pass through, such as the oxygen, the carbon dioxide, and the steroid, which dissolves in hydrophobic bilayer, and it undergoes simple diffusion. And other substances such as the sodium chloride glucose and hydrogen, those are water soluble. This uses channels or carriers such as integral protein. So in cell membrane, the water is lacking. So lipid soluble substances can pass through or undergo in simple diffusion. So what is lipid bilayer? Lipid bilayer is composed of what we call the fluid mosaic model, and it consists protein, phospholipids, cholesterol, other lipids, and carbohydrates. So for the protein, it is 55%, and we have in the lipid bilayer, the protein we have two types: the peripheral proteins and the integral protein. For the peripheral protein, this is electrostatic interaction, and it has a loose attachment, and it controls the transport of the substance, and it functions as an enzyme. So this protein, the peripheral protein, is the reason for the fluid mosaic appearance. Another type of protein is the integral protein. This acts as a carrier proteins, and it provides structural channel or pores. And this is hydrophobic interaction, and it has a tight attachment. And that's the two types of the protein in the lipid bilayer: the peripheral proteins, which have loose attachment, while the integral protein have the tight attachment. And it functions as a carrier, and the peripheral proteins function as an enzyme. And aside from proteins, we also have the phospholipids. This is the fatty acid portion, and it also have the phosphate end. So for the fatty acid portion, this is the hydrophobic part, and it is non-polar tail, while the phosphate end, which is the glycerol, it is the hydrophobic polar head. So that's the phospholipids. It contains fatty acid portion, hydrophobic non-polar tail, while the phosphate end or the glycerol is a polar head. And aside from the phospholipids, which is composed the lipid bilayer by 25%, we also have cholesterol, which is 13%, and this cholesterol reduces the fluidity and prevent the solidification. It controls the fluidity of the cell membrane, and cholesterol also helps to determine the degree of permeability of the bilayer to the water and soluble constituents of the bloody fluids. And aside from cholesterols, we also have other lipids which compose the lipid bilayer by 4%. And next is the carbohydrates, which is 3%. And the carbohydrates are entirely outside the surface of the cell and has a loose carbohydrate coat, which is glycocalyx. So this glycocalyx is the loose carbohydrate coat, and it is highly hydrophilic, and it protects the cell from ionic and mechanical stress. Okay, so that's all for the lipid bilayer. Now let's go back to the cell membrane, and let's remember that the cell membrane divides the body to the extracellular fluid and intracellular fluid compartments, and it is more permeable to potassium rather than sodium. Okay, so aside from the lipid bilayer, the cell membrane also contains the intercellular structures, and we have here the macula adherens, or also known as the desmosomes, and its function is for tight intracellular adhesion, and it is found in the epithelium, and it is like staple wires. So another types of intercellular structures is the zonular adherens, and this increase the surface area for contact, and it is found at the epithelial and endothelial and cardiac muscle. And the zonular adherens is like a fascia adherens in the cardiomyocytes. And aside from the macular adherens and the zonular adherens, we also have the zonular occludens, or also known as the tight junction, and it serves as a barrier to movement of proteins. And these zonular occludens or tight junctions divides the cell to an apical and basolateral. And it has two types: the tight junction, which is the impermeable, and the leaky zonular occludens, which is the permeable. So the tight junction, it is found in the CD and terminal colon and BBB or blood-brain barrier, and in the renal distal tubule. However, the leaky permeable is found in the proximal tubule and in jejunum and gallbladder. Another intercellular structures between the cell membranes is the gap junction, and this junction is for rapid intracellular communication. It is found in the cardiac and uniform muscle. And the gap junction function unit is known as the connexin, and the subunit is the connexin. The functional unit is the connexon, and the subunit is what we call connexin. An example of these is electrical coupling between the myocardial cell. So that's the gap junction. It is used for rapid intracellular communicator, and its functional unit is known as connexon, and the subunit is connexin. And this junction has what it's called syncytiums, or the cardiac muscle, and there's unitary muscles that contract together, so that's syncytium. Now let's discuss the transport across the cell membranes. So first we have two types of transport: the non-carrier mediated and the carrier mediated. So first, let's tackle the non-carrier mediated. We have simple diffusion, endocytosis, and exocytosis. So for simple diffusion, this does not require metabolic energy, and it transports substances from high to low concentration, or what is known as downhill transport. So example of the substances includes the oxygen, the nitrogen, carbon dioxide, the alcohol, lipids, hormones, and anesthetic drugs, as well as the lipids, the cortisol, the fat, soluble vitamins, and the thyroid hormones. So that's the lipid hormones. So these substances uses simple diffusion, and simple diffusion have an equation which is this one: the flow, and the flow is equals to the permeability times the area, and times the concentration 1 and concentration 2 in moles per liter. And this permeability here is the factors that there are factors that increase the permeability of the membrane. So the factors that increases the permeability includes the oil or water. There's increased oil and water partition, the water partition coefficient. So that's a factor in increasing the permeability if there's an oil or water partition coefficient. So it means that the substance is more lipid. And if there's a decrease in radius or size of the solute, so it is more permeable if there's a decreased size of the solute or decreased radius of the solute. And it is also more permeable if the membrane thickness is decreased. So that's the three factors that increases permeability. If it's more solid, if it's more lipid, if it's more decrease in size or more smaller solute, and if the membrane is thin. Okay. So another type of the non-carrier mediated is the endocytosis. This means that it is the ingestion of the cell or ingestion by the cell, which have two processes, the pinocytosis and the phagocytosis. The pinocytosis is most of the large macromolecules such as protein molecule can enter the cell. And in phagocytosis, so pinocytosis is like drinking, cell drinking. However, the phagocytosis is like cell eating. So it involves particle rather than molecules. So in pinocytosis, it's macromolecules. However, in phagocytosis, it is particles. And it's only certain cells have the capability of phagocytosis, such as the white blood cell and macrophages. So white blood cell and macrophages has the only cells that have the capability of phagocytosis. And there's also the process what we call opsonization. Opsonization is the intermediation of the antibodies. Now let's go to another type of the transport across the cell membrane is the carrier mediated. So this transport uses a carrier, which are the osmosis, facilitated diffusion, primary active transport, and the secondary active transport. Okay, so first let's discuss the carrier mediated. What are the characteristics of carrier mediated? It includes stereospecificity, saturation, and competition. Stereospecificity. Example is there may be two substances that may have same chemical composition but different shape, such as in D-glucose versus the L-glucose. So they have same composition but different in shape. And this stereospecificity is the carrier accommodates specific shape. Another characteristic of carrier-mediated is the saturation. So the transport rate increases as the concentration of solute increases until the carriers are saturated. And the numbers of carrier are finite. Once all carrier are saturated, rate of the transport become constant. And the transport maximum is analogous to enzyme kinetic maximum velocity. So transport maximum is an analogous to enzyme kinetics maximum velocity. So that's under the saturation of the carrier-mediated transport characteristic. And another is the competition. It means that the solute compete for transport sites on carriers molecules. Example is the galactose. Galactose is competitive inhibitor of glucose transport in the small intestine. Like there is other transport. Yeah, the SGLT and the SGLT type 1. Okay. So these are the three characteristics of the carrier mediated. It is stereospecific. It means two substances may have the same chemical composition but different in shape. And saturation, the transport rate increases as the concentration of solute increases until the carrier are saturated. And the competition, which means the solute competes for transport sites on carrier molecules. Okay, so now let's go to the different types of the carrier-mediated. First, we have the osmosis. So in osmosis, the flow of water across the semi-permeable membrane is from the low concentration to high concentration. A while ago, in the simple diffusion, it's an opposite. It's from high to low. However, in osmosis, it is from low concentration to high concentration. And the osmolarity, we have here what we call the osmolarity. It is the concentration of osmotically active particles in solution, and it's the property that can be measured by freezing point depression. And the equation for the osmolarity includes the number of particles in solution times the concentration in the ECF and ICF osmolarity is 300 milliosmol per liter. And the osmotic pressure can be calculated by Van Hoff's law. So it depends on concentration of osmotically active particles. And the concentration of particles is converted to pressure. So the osmotic pressure is equals the number of particles in solution times the concentration times the gas constant and the absolute temperature. This is the concentration of particles that converted to pressure. So it is number of particles times the concentration times the gas constant, which is 0.082 liters at millimole, and the absolute temperature. Okay, so under the osmosis, there's also what we call the reflection coefficient. This is the number between 0 and the one describes the case with which solute permeates on a membrane. So we have here, if the reflection coefficient is 1, the solute is impermeable and it creates osmotic pressure and it causes water flow. So example of those who have reflection coefficient 1 is the albumin. So it is impermeable and it creates osmotic pressure and causes water to flow. And if the reflection coefficient is 0, so the solute is completely permeable. And example of these are the urea, and it is an ineffective osmol, the urea. So it has 0 reflection coefficient. So reflection coefficient is measured by the amount of the return over the amount that is sent. Aside from the osmosis, we also have the facilitated diffusion, and it is like the simple diffusion that moves the substance from high concentration to low concentration, or also downhill transport. And it is passive, but it is more rapid than the simple diffusion. So if the solute is low concentrated, the facilitated diffusion is far faster than the simple diffusion if it is low solute concentration. But if it is high solute concentration, the facilitated diffusion is slower than the simple diffusion. Example of the facilitated diffusion is the AA transport, or the amino acid transporter, and the GLUT transporter. So for the GLUT transporter, we have type 1, 2, 3, 4, and 5. For the type 1, GLUT transporter can be found in the BBB, in the placenta, in the RBC, and the cornea. And type 2 GLUT transporter can be found in the liver, pancreas, or in the basement membrane of the small intestine and the kidney. And that GLUT3 can be found in neuron and as well in the placenta. And the type 4 GLUT transporter are found in muscles, adipose, and remember that this GLUT4 is the only insulin-dependent glucose transporter. And GLUT5 is a fructose transport from small intestine lumen to small intestine cell, and it is spermatocytes, found in spermatocytes, the GLUT5. So that's are the example of the facilitated diffusion. Now let's go to primary active transport, another carrier-mediated transport that transport the substances from low to high concentration, same as the osmosis. And yeah, it is uphill, like osmosis. And at its name, it's primary active, it is active, and it exhibits co-transport, or what we call the symport, and the counter-transport, which is known as the antiport, or exchange. So the source of energy of primary active transport is the ATP hydrolysis. So that's the primary active transport source of energy. Here are the examples of the primary active transport, such as the sodium-potassium ATPase pump. Here there are 3 sodium going out and 2 potassium going in. So 3 sodium efflux and 2 potassium influx. And this sodium-potassium ATPase pump contributes to RMP of 4 millivolts out of negative 70 millivolts. And it contributes to BMR, and it inhibits by the digitalis, or what is known such as digoxin. And aside from sodium-potassium ATPase pump, we have here the calcium ATPase pump, and it is found in the sarcoplasmic reticulum, or SERCA, and it pumps calcium back to the sarcoplasmic reticulum, the SERCA. So that's the calcium ATPase pump. And another example of the primary active transport is the hydrogen potassium ATPase pump, and it is known as the proton pump. So that's the primary active transport. Now let's go to the secondary active transport. And this moves the substance from low concentration to high concentration, also an uphill transport. And it is also active and it exhibits co-transport and counter-transport, same with the primary transport. And in the co-transport, or symport, it moves the solutes in the same direction, and the counter-transport or the antiport exchange, it moves the solute in the opposite direction. So in the example of the symport or the co-transport is the SGLT. It moves downhill, the sodium moves downhill, and the glucose moves uphill. So sodium downhill from high to low concentration, and the glucose moves uphill from low to high concentration. So here the SGLT1 in the small intestine can be seen, and the SGL2 can be seen in the kidney. Another example is the sodium potassium chloride co-transport. So this is found in thick ascending limb, and it is inhibited by the loop diuretics, the sodium potassium chloride co-transport. And another type of the secondary transport is also the co-transport or the antiport exchange. So the example of this one is the sodium calcium exchange, and it is found in the cardiac membrane. It lowers the intracellular calcium, and aside from sodium calcium exchange, another example of the counter transport is the sodium hydrogen exchange. So the source of energy in the secondary active transport is the downhill transport of the sodium. It relies on the sodium potassium ATPase pump. So remember that it relies on the sodium potassium ATPase pump. So that downhill transport of the sodium. So the sodium is from high concentration to low concentration. So that's about the source of energy of secondary active transport. So that's the transporters of the cell membrane. And before we go to the cytoplasm and organelles, we'll have first trivia here. So there's 68 trillions of human cell plus bacterial cell, 30 trillions of human cell, 1 trillion of glial cell, and 100 billion of neurons. So there is 1 to 10 ratio between the glial cell and the neurons. So 1 trillion glial cells is equivalent to 100 billion of neurons. So what are the factors that determine permeability of the cell membrane? It includes the temperature, the type of solute, and the level of cell dehydration. So if the temperature it is high, it is more permeable. If the solute is lipid, it is more permeable. Now let's discuss the other parts of the cell, which includes the cytoplasm and the organelles. So the cytoplasm is composed of what we call the cytosol. It is a jelly-like fluid portion of the cytoplasm in which particles are dispersed, and it is mainly dissolved proteins, electrolytes, and glucose. So that's cytosol. Another is the nucleus, and this is the control center of the cell. It contains the DNA without comprise the genes. We also have the endoplasmic reticulum. This helps to process molecules made by cell and transport. And it transports to specific destination, the molecules. And there's two types of the endoplasmic reticulum, the rough and the smooth. So the rough is attached to the outer surface ribosomes composed of the RNA. So the rough endoplasmic reticulum contains ribosome, and it is found in the outer surface. It is composed of RNA and proteins, the ribosome, and the function of this is to synthesize the new protein. And remember that the rough endoplasmic reticulum is also known as granular endoplasmic reticulum. And another type of endoplasmic reticulum is the smooth endoplasmic reticulum. Smooth meaning it's granular. And this smooth endoplasmic reticulum synthesizes lipid and degradation of the other process of the cells. So it synthesizes the lipid, the smooth endoplasmic reticulum, and it's promoted by the interreticular enzymes, and there's no ribosome attached in the smooth endoplasmic reticulum. Another function of the smooth endoplasmic reticulum is it stores calcium ion. It's a calcium ion storage, and it breaks down toxins in the liver. Another content of the endoplasmic reticulum is the endoplasmic matrix. It is a space inside the tubules and vesicles, and it is a watery medium. Another organelle is the Golgi apparatus. It is prominent in secretory cells, and it functions associated with endoplasmic reticulum. And the Golgi apparatus is the transported substance are processed in this Golgi apparatus to form lysosomes and secretory vesicles and other cytoplasmic components. So the function of this is it transported substance are processed to this, and it forms lysosomes, secretory vesicles, and other cytoplasmic components. And it packages cellular products in vesicle, the Golgi apparatus, and it is known as the central delivery system of the cell. So that's Golgi apparatus. Another organelle is the lysosome, and it is a vesicular organelle with 250 to 750 nanometer in diameter, and these lysosomes provide intracellular digestive system. It is the major digestive unit, the lysosome, and if it gets damaged, it causes lysosomal storage disease. So again, we've discussed that lysosome provide intracellular digestive system. It allows the cell to digest. So first, it damages cellular structure, and number two, the food particles, and number three are the unwanted matter such as bacteria. So that's what allows the cells to digest these three things: the damaged cellular structure, the food particles, and unwanted matter such as bacteria. The lysosome is surrounded by lipid bilayer membrane, and it is filled with a small granules like 5 to 8 nanometer hydrolase or digestive enzyme. So hydrolase can be found in the lysosome. It is small granules, and this is the protein hydrolyze the amino acid. So that's the hydrolase, and glycogen to glucose, and lipid to fatty acid and glycerol. So that's the function of hydrolase. So aside from lysosomes, we also have what is called peroxisomes. So it is formed by self-replication, such as budding off from the smooth endoplasmic reticulum. So peroxisomes contains oxidase to form hydrogen peroxide, and it is associated with catalase. So peroxisomes contains oxidases. It's form hydrogen peroxide that's associated with catalase. And peroxisomes, another function is it catabolizes long chains of fatty acid. Another organelle is the mitochondria. It is known as a powerhouse of the cell, and it synthesizes ATP. It's self-replicative, and it contains DNA, the basic chemical in nucleus that controls replication of the cells. So we also have cytoskeleton. This is a fibrillar protein, and cytoskeleton helps to maintain the shape and allows the cells to move. So we have here three elements: the microtubules, the microfilaments, and the intermediate filaments. So the microtubules is the one that forms the centrioles and the mitotic spindles. So that's the three elements under the cytoskeleton. So I think we've discussed the cytoplasm and the organelles well. We have again the nucleus, cytosol, endoplasmic reticulum, which composed of rough and smooth endoplasmic reticulum, the Golgi apparatus, the lysosome, the peroxisome, the mitochondria, and the cytoskeleton. So that's all about the cell, the basic unit of human body.